I don't have anybody. I'm unaware of anybody that's for sure not going to be here, but it's pretty unobtrusive to just run the recording back over there. So I'll just go ahead and record, and that way, I don't know, maybe it helps you guys with your studying later. The other thing is tonight, as we've mentioned, we'll start a topic that doesn't show up until test 4. We'll start chapter 12. We'll take test 3 on Monday. That gives you the weekend to focus on your studies for chapters 9, 10, 11. Okay. That doesn't put me out any to back that up, but it does mean we got to start a topic that's again not on test 3. This stuff will show up on test 4. So I don't mind recording it, and that way, you know, it's going to be a couple weeks before we take test 4. You can go back and review this later. Okay. Well, with that said, just as normal, we are recording, so let's not mention any names, but feel free to ask your questions. Okay. We'll do labs 8 and 9 tonight. Good news, these are labs that are along with the material from chapters 10 and 11. This is mitosis and meiosis labs tonight. So we've gotten through the labs that was material that we've already tested over. Now we'll start labs on material that we're working on right now. So that's good news. Quickly on, I goofed. Right? And I made a mistake. If we go look at your modules here and we go look at the lab module. So the last minute as I was adding lab 8 and 9 and their respective quizzes, and I noticed lab 6 and 7 quizzes were not published. Oops, that's my fault, right? Lab 6 and 7 quizzes, you couldn't take them, so I'll give you a couple more days to take those. Normally I would make those due at midnight, the day of the lab, right? So there's lab 6 and 7 quizzes. Those are, and to even sweeten the pot further, I gave you 2 attempts at those. Keep your highest attempts. Now, I do that. I understand that. There's like a set number of points, and if I miss a number of points, then I got to figure out. It's much easier to just give you some extra time than it is for me to try to figure out, okay, where am I going to add points now? Yeah, so you got until Monday. That'll be after the exam. I would suggest, because those are, let's see. Yeah, just get those done. It wouldn't knock them out probably this weekend. You might be able to knock one of them out before we get lab started here in a little while. Anyway, knock them out. You got time there. And again, to make it even better, I gave you 2 attempts. So I don't plan to do that moving forward, but I didn't mind doing that here because I made a mistake. Okay, questions there? Makes sense? Clear? Now, lab 8 and 9 are published because they're in green. They are published. The lab quizzes for 8 and 9, so those will be your normal time. Those will be standard procedure. Okay. All right. What else here? I think those are the main things. I did create Unit 4 materials for Test 4. That's where tonight's lecture is. After Test 3 is done, I'll move this one up to the top. And so he'll be up here. Unit. That's awesome.
We went over lecture 17, test 3 review, right?
Yes, we did that Monday.
So that's tonight.
That's down here. There's lecture 18, chapter 12. Okay. All right, what else? Okay. How's everybody doing?
Fantastic.
I like it. See some thumbs up, some thumbs down. I get it.
Cinnamon rolls today.
I had a cinnamon roll today.
I made them.
Oh, yeah, well, yeah, no, I just bought one. My wife got one, so. Yeah.
What's the good APR rate for an auto loan?
Oh, man. In today's market, if you got good credit, I wouldn't pay more than 4% right now, but, you know.
Right? You think you're at 4%?
4.1. Come on. You know, come on. It's not straight.
Hey, honestly.
And if it's below that, then I need to switch to Navy Federal or wherever you're banking.
I get it. It's been a while since I priced one. If you get anything less than 6%, you're doing pretty good on a car loan, honestly.
Right.
So, yeah. All right. Any biology-related questions or course-related questions?
My friend did a terrible idea.
I wasn't specific. That's fair. All right. Let's rock along. So again, test on Monday. I called over at the office. It appears ET201, the computer lab upstairs, that it should be available. So just have yourself where we can get the Canvas, and we'll take the test there on Canvas and That way it's, we know how I am about grading, so that way it's, most of it's already graded for you, so. And I'll go through and look at all the fill in the blank ones and make sure that if you've missed some spelling or something like that, that's no problem. I will tell you, you're later, for most of you going into biological and medical fields, your later courses, they will start getting very, very picky about spelling, even the really technical stuff. I'm very forgiving here because I'm not a biologist, I'm an engineer. Right? So I'm very forgiving about your spelling. I'm just telling you, if this is the direction you're headed, if you're headed down the biological realm, you've got to get really good at your spelling. They will be very picky about that later.
Are you forgiving about the math and asterisk notation?
In here, yeah, I would be. Yeah, if you're in one of my later engineering courses, no, that's different. That is our language. We're not as forgiving on that. Yeah. Okay. All right. Again, we'll start chapter 12 tonight. This is my subject. The reason I'm so strong in biology is because, again, I thought I wanted to be a genetic engineer at one point in my life, and I still toy with that idea, honestly. But tonight we're going to learn about one of the fathers of genetics. Essentially, the man is credited as the father of genetics, modern genetics. And we'll look a little bit about how he figured out what he figured out and what that means for us. We're going to study Mendelian genetics tonight. Gregor Johann Mendel was born Johann Mendel. Did a little digging on him today. Lived in the 1800s. He was a devout Catholic Christian. He sought support from the church so he could further his academic studies. So he basically became a monk so that he could utilize the academic resources and capabilities of the church. He was in Europe, Austria area, but he was a man of science through and through. A physicist, biologist, mathematician. He liked to teach when he could, but when you look at the work that he did, it was very much lots and lots of scientific experiments, interested in meteorology as well. He came across and founded essentially keep in mind, in this time frame, he figured out how genes were passed from one generation to another, or at least a lot of the foundational information we need to understand. Keep in mind, we did not necessarily at this time frame know what DNA even was. We weren't even sure what molecules were sometimes in this time frame. We understand today that the genes are passed through nucleic acid molecules. But again, that even to Mendel, it's probably going to be, I don't know if we talk about nucleic acid and passing on genes, that's not the language they would have used. All right, so really forming a foundational basis. Now, in his time frame, this was the prevailing theory as to how genetics occurred, how information was passed on from one generation to the other. And the idea was, based in observation, in populations, was this blending inheritance. This was the prevailing theory in the 1800s in biology as to how characteristics are passed on. Okay? And so you can see there, and I just pulled some information off Wikipedia. I saw this graphic. I was like, that's perfect. Let's borrow that from Wikipedia. So the idea there is we're just saying a simple example of flower color. They're saying if you start out, you crossbreed red and white flowers together, the next generation is going to have a lot more pink flowers, some red, some white flowers, mostly pink flowers. And the next generation after that is going to have more pink flowers, some reds, and then the following generation after that is just going to have all pink. So it's just this idea that you inherit half of your characteristics from each parent, and they just get blended with each subsequent generation. Now, that can happen depending on what genes and what traits we're talking about. That can happen where you put two two donors of genetic material together, and their offspring will have some appearances, a blending of appearances of both. So it's understandable why this would be the prevailing theory, but this was the prevailing theory. What we have to understand is Mendel conducted experiments, and the results he found did not support the prevailing theory. Okay? So imagine, and again, keep in mind, we're talking about the 1800s here. We're not talking about modern time. We see today with the idea of climate change, where when you look at the evidence and the scientifically researched data that supports the idea that as we increase greenhouse gases in our atmosphere, we will observe climate changes. That's the science on that's pretty solid, and it's pretty easy to see how much support there is to discredit that information. That's in modern times where we have access to correct information, readily available access to correct information. So put yourself in this poor guy's shoes. He's figured out something that goes against what the accepted prevailing theory is, and it's in the 1800s. So his findings didn't really come to light until after his lifetime, early 1900s. Part of that's just communication of information. It's slow in this time frame. The scientific community is still growing and maturing in this time frame. But again, just kind of keep that in perspective. He went on later, as you do a little research, he went on later in his life to become abbot within his church, and he found himself, he wasn't able to conduct research and continue his findings later on in life. He was kind of bogged down with the administrative challenges of operating a large church. Okay? But hey, we still appreciate what he was able to do for science. Okay. So I pulled this image from your text that on the right, again, just to kind of show you. We see here what on the right. We see here what Mendel found is that if you took purple flowers and white flowers and combined them, the next generation he would get purple flowers, and then the subsequent generation he would get a mix of purple and white flowers. Which again, that goes against the prevailing theory at the time, where you cross these enough times and you're going to get just this blurred blend of the two. And what he found is, and he did this for multiple traits in pea plants. We refer to the Mendel and his peas that he conducted experiments on. So just green peas that you would eat, all right? Similar, in the text mentions the exact species of the peas. He conducted approximately 24,000 individual plants, experiments with 24,000 individual plants. Working in a monastery, he was a gardener, enjoyed gardening, they had a large garden, and he was able to dedicate a lot of his time to meticulously ensuring that individual pea plants were only reproducing with genetic material from other pea plants that he wanted to be able to control and test and observe specific traits. So he studied over the course of 10 years, he studied 7 different traits in pea plants. Again, about 24,000 plants. So again, just try to imagine the scale. If you just take a moment and think about the scale, how much, whew, that's a lot. That's a lot. He's looking at the shape of the seed. Is it round or is it wrinkled? Color of the seed, yellow and green. Flower color of the flowers, purple and or violet is often what we'll call it, violet and white. Different types of seed pods. So there's this inflated or the constricted, where it's inflated. It's kind of like if you see snow peas or have eaten snow peas. It's kind of a big wide pod. Constricted being where the pod kind of hugs the individual seeds. Yellow or green. And then some arrangement and position of the flower. And we don't necessarily have to get into the technicality. It's not a botany class here. I'll mention a little bit about plant reproduction just in a second so we understand some context. But some arrangement within the flower, and then also the height of the plant. We call kind of the normal or tall plant versus a dwarf or a shorter plant. So again, these are the 7 traits he studied over 10 years in this particular pea plant, trying to determine how they, how do they pass genes on from one generation to the next? And let's see here. Give me a second here. No? Oh, enable editing. Okay. I apologize. I wanted to be able to draw on the slides as we go, and I've got to go through a couple steps to do that with this smartboard for whatever reason. So, now I got to wrestle with that. Okay, now I should be able to draw. Apologize for the delay. Okay. So, again, not a botany class. I'm not a botany expert. I'm not going to pretend to be, but we got to understand the basics. And to be able to talk about what the heck Mendel was doing, we need to understand a little bit about how flowers work. So if you've not seen this before, most of us have probably seen this before, but if you haven't, flowers have both male and female reproductive parts within them. Most flowers do, not all. Some species will have male flowers and female flowers. Sometimes entire plants will have only male flowers, entire plants will only have female flowers. Sometimes they'll have them at different locations, right? Corn plants, for example, have male parts and female parts at different locations on the stalk. But when we see a flower, most of the time there's going to be kind of the central structure, the stigma. This is where the pollen will come from the male parts, right here on the stamen. Little pollen particles that carry the sperm will come and, whether a bee comes along and picks it up, or whatever the case may be, or the wind blows it around, those pollen particles will stick to this female portion, and then that will deliver the sperm cells down here where the egg is, and fertilization will occur. So similar to what we see, again, gametes from the here will come over and combine with gametes in here. Similar to what we see in other species, a little different. Again, there's both male and female parts in most flowers of most plants, meaning they can self-fertilize. And what's important we have to understand here, in the particular pea species that Mendel was observing, the flowers would often fertilize themselves before the flower would even open up and allow, where most flowers will open up and they'll allow then, again, pollinators like bees or something like that to come in and spread the pollen around. Or wind can carry in pollen from other plants, and we get crossbreeding. The peas that Mendel was working with almost exclusively would self-fertilize because they would often fertilize before the flower would even open. So they would fertilize only with pollen contained within the flower. So it's essentially inbreeding, which is, for in animal species, that's hard for us to understand because in all animal species, we know that in general inbreeding tends to be bad in many cases, but in plants it's a little different. In general, a lot of plants will actually inbreed, self-fertilize, and to the extreme inbreeding. That's okay. For plants, that's just fine. So what Mendel did is he would actually go in and he would remove the male parts from the plants that he wanted to fertilize with pollen from other plants, the ones he wanted to crossbreed. He would actually, before the flower would open and self-fertilize, he would go in and actually manually remove these stamens from inside the flowers before they had a chance to self-fertilize. All right, so I've got kind of what I've done in the next few slides here. I've got a set of rolling terms. So basically, when the page fills up, the terms will move up. So feel free to write these as we go along. When I've filled the slide up, don't panic. The latest terms are still going to be on the next page. Okay. A term that I was not familiar with personally, that and as I was reading through the text, and I saw this term kept popping up, and I guess I didn't catch it the first time they defined it. I had to go back later and pick this up. The term true breeding, and that's what we're talking about with the peas that Mendel was working with, is that's where the term true breeding simply tells us that the offspring will always look like the parents. Okay, the offspring will always look like parents. I don't know why I won't have parents up there. Missed that. That's going to show up on several slides, but that's okay. So the offspring will always look like the parents in a true breeding situation. With the peas that Mendel was experimenting with, they were always true breeding because often they would self-fertilize themselves. So the offspring would basically be clones, well, not entire clones, but much like clones of the parent because it's themselves. But what that means is that if you have purple plants, purple flowering plants breeding with purple flowering plants, you're going to have offspring that appear to be purple flowering. That's a true breeding pair. You have black cattle mating with other black cattle, they will produce black cattle. It's true breeding pairs. Now this naturally occurred in the pea plants Mendel studied since they bred themselves. It's mentioned that, okay? Now, so again, I think these terms are just going to stay there. I'll bring in some pictures and some images here and there. They'll kind of come and go, but the terms will stay. So I took a direct quote from an Oxford dictionary on gene. So I thought this was better than I could put together. And it took me a minute to kind of, I never found exactly what I was looking for in the text. So I'll just pull a definition. And then as I did, I saw this cool graphic from genome.gov, and I thought I would show that too. It's a beautiful graphic for what is a gene. A gene is simply a strand or a portion of a DNA strand. It's a distinct sequence of nucleotides forming part of a chromosome. It's also it can also be in genes within viruses. Right now, if you've received the Pfizer or the Moderna vaccine, you basically have received an mRNA molecule or a bunch of mRNA molecules that codes for the gene of the coronavirus spike protein. So it's the protein on the surface of the virus that attaches to your cells and allows that virus to then invade your cells and use your cells to create copies of itself. What Pfizer and Moderna have done is they've figured out how to create and package an RNA sequence that your body can then use to build copies of that spike protein, just the spike protein. So it's not the whole virus. It's just the weapons that virus used to attack your cell. And so basically, we're giving your cells the blueprints for the weapon that's going to be used against them. So when your cells then encounter that weapon, if the coronavirus happens to try to attack your cells have already seen the blueprint for what the weapon looks like and has already started producing thousands and millions of copies of this weapon. And then when your body says, wait, what am I doing with all these spike proteins laying around? I don't need those. And so your body figures out how to destroy those spike proteins. So then when the coronavirus comes along with the spike proteins, your body has already figured out how to destroy those spike proteins because we've given it that gene. All right? We've given it that gene, the sequence, the set, the distinct code, DNA snippet that contains the information. That is what a gene is. Now, an allele is a different version of the gene. So there can be different versions of a gene. For example, blonde hair or dark hair, or I'm trying to think of some other simple genes here. I mean, for example, with seed color, yellow or green seeds, those are different alleles. So on a particular homologous chromosome, at a particular site on one of the chromosomes will be the gene that will code and determine the color of the seed within the pea pod. And so the question is, well, is it a green allele or is it a yellow allele? And that will help determine whether we produce yellow or green seeds. Now, these are closely related to the trait. The trait would be the color of the seed in this case. Right? The trait is the physical appearance or expression resulting from the alleles present for a particular gene. So So I've got a bunch of terms here. I looked at the way the chapter presented the material, and they've just kind of spread these terms out. And I'm like, between a couple of sections, I'm just like, these are all terms. We need to load this up. Let's get the terms done the first day. And then we'll probably do a second day on chapter 12 after the exam, and we'll kind of refine the terms a little bit as we go. But let's get you guys loaded up with the terms here. I'll write these here as we go. All right? Terms. I'm not going to write the definition. I'm just going to write the terms down. True breeding. So you can point back to them and say, we've already defined this. True breeding, gene, allele, trait. Okay, so just going to keep a running list of the terms. Now again, so what I'm going to do is I'm just going to keep adding terms here, so I'm just going to move the other terms up. So if you miss one, that's okay. It'll still be on the next slide. And just the top one gets rolled off as we move forward, okay? So there are two types of traits that we can encounter. There's a dominant trait. So you're going to get two alleles, one from each parent, right? You have pairs of chromosomes, pairs of homologous chromosomes. Each homologous chromosome contains the same genes on each chromosome. Now, you're going to get a set of alleles on one chromosome from your father, one set of alleles on the homologous chromosome from your mother. They may or may not be the same alleles. One might be for yellow seeds, one might be for green seeds. Humans aren't going to have that, but plants do. Right? The dominant trait is the one that will get expressed if at least one allele is present. So it doesn't matter if you've got two, I think it's green is the dominant, so I think if you've got two greens or one green, if green is the dominant trait in the alleles, that's the one that get expressed if either the one from your mother or the one from your father contains the dominant one. The other type of trait is the recessive trait. Recessive trait. I'm left-handed. Now, we're not for sure whether left or right-handed is determined by a single gene in humans. We don't know that for sure. But if it is determined by a single gene, then the only way I'm going to be genetically left-handed is if I received a left-handed allele from my father and a left-handed allele from my mother. Okay. What's that?
My brother's left-handed, and all of his brothers, they're not mine, they're not related to me, but all of his brothers, his 7 brothers, 1 sister. Sister's right-handed, but all the boys are left-handed.
Interesting. And again, we're not sure if for a number of reasons, we're not sure that it's going to be beyond our scope. We're not sure if left or right-handed is a single allele trait in humans. It's possible it's controlled by more than one allele, and then the genetics get a lot more complicated. It's harder to describe that. There's also a cultural aspect about left-handedness. For a long time, we have forced kids to be right-handed. It's only been the last century or so that it's even been acceptable to allow your child to be left-handed. Today, it's like weird to us. Why in the world would you force that kid to do that? Right? We know it's going to cause lots of mental problems later. Anyway, it's possible it's single. We're not sure. We have to figure that out. So again, a recessive trait requires both alleles to be for the same trait in order for it to be expressed. Now, what that means then is you have what we call the phenotype. The phenotype is the traits, essentially it's the traits that actually get expressed. That's the phenotype. So So generically, it's just what do you actually see or observe when you look at that individual. That's the phenotype. And right now, these are just terms. We're going to try to put some context to them as we go. And so it's okay if it doesn't make sense yet. Just defining them right now. Phenotype is just basically what do we observe? What's observed in that individual? As opposed to the genotype, so the genotype is what's the actual genes, what are the actual alleles that individual carries. Because again, remember the dominant trait can overpower the recessive trait. So if there's one dominant trait and one recessive trait, or I'm sorry, one dominant allele and one recessive allele for the same trait, the dominant will get expressed. So we'll observe the dominant gene, even though the genotype level, genotype is they've got one allele of each. We only see the dominant one, the one that's being expressed. And so right along with that, types of genotypes we can observe you can have a heterozygous genotype or you can have a homozygous genotype. So homozygous genotype is where you get the same allele from each parent. So one gene, you get two different alleles, one from each parent. Same location on homologous chromosomes. If it's homozygous, that means you got the same information from your mom and your dad. So that's the one that will get expressed. Heterozygous means you got different alleles from each parent. So then it comes down to, okay, which one's dominant? That'll be the one that gets expressed. So again, the genotype can take two flavors. Either the same allele or different alleles. Okay. And then graphically. Okay. So I misspoke earlier. Green is the recessive one. Wouldn't have thought that. At least in this particular pea species that Mendel was doing his experiments with. Green is the recessive type. So here, and what we'll often do, in a lot of genetics, so we'll say if we're describing whatever gene we're describing, whatever trait we're describing, whichever one is dominant, we'll put the capital letter. So now we'll communicate this information. We're talking about alleles for a gene, or alleles for a trait. Whichever one of those traits, whichever trait is dominant, we'll use the capital letter for them. We'll use the lowercase of that same letter, even though it's green. If the yellow allele is dominant over the green allele, we'll use a capital Y for yellow. We'll use the lowercase of the dominant trait, lowercase y, to describe the recessive trait. So in the case of the particular species of peas that Mendel was studying, the yellow color was dominant over the green color. Now the yellow could occur a yellow allele from each parent, or it could also occur with a yellow allele from one parent and a green allele from another parent. So we call a homozygous where you have two of the same. Heterozygous is where you have one of each. So if we're displaying the dominant trait, it could be homozygous for the dominant trait. It could be heterozygous for the dominant trait. There's just looking at it, we don't know. Modern times we've figured out some ways. A lot of times we can, if we can figure out where on which chromosome this trait is coded, a lot of times we can go in and cut that portion out of the chromosome and study that and actually do some tests and figure that out. I think I mentioned before, I grew up showing livestock. And I grew up showing cattle, raising cattle. At that time frame, solid black cattle, certified Angus beef was really, really important in terms of marketability of those animals. So there was a heavy push, because black is dominant over red in terms of genetics. There was a heavy push, and they actually devised genetic tests where you could send in on a bull, you have the bull tested, is he homozygous black, so every single offspring he produces is black, or is he heterozygous black, and he's more valuable to us if he's homozygous black, just for example, right? So there's sometimes that dominant trait, we might actually, because we can't just look at him and see, we need to be able to test him. And if there's a market reason to do that testing, science will figure out how to do that testing, okay? I don't know in peas if there's enough value involved with having yellow peas that we would actually see, well, is it heterozygous or homozygous? But if it's displaying green, the only way it's going to do that is to have a green allele from each parent. So if it's displaying green and green is the recessive trait, then we know that plant, right, if this is my phenotype, it's either yellow or green, if the phenotype is green, then I know the genotype has to be homozygous green. That's what we're saying here on this graph. Homozygous dominant or heterozygous dominant will both show the dominant trait. To show the recessive trait, it has to be homozygous with the recessive trait. Okay. Questions so far? That's a lot of terms to throw at us very quickly. Okay. Again, Mendel studied 7 traits in his peas. Just kind of quick review here. Now, the conclusions he came to, and again, remember, everybody around him at this time thinks, oh, well, you just cross enough yellow and green peas together, and eventually you'll get nothing but these kind of yellow-green cross peas, right? It's some mixture of yellow and green is just what you're going to get out of the seeds eventually. And Mendel knew from just kind of some basic experiments, he knew that's not what he observed, right? And he wanted to try to understand why. So he devised experiments where, again, he could control which plants bred with which plants. And he tried to figure out if he could measure how the genes or how these traits were being passed from one generation to the next. So we just crop that out. Let's just talk about the flower color first, which I think in your text is what they start with as well. We just talk about the flower color first. So this is what Mendel did, is he started with what we call the P generation, the parental generation, or the parent generation, up at the very top over here. He took violet flowers, or purple flowers, if you like, and crossed those with white flowers, knowing that if you just left the purple flowers to breed with purple flowers, you get purple flowers, and just let the white flowers breed with white flowers, they get white flowers. But what happens when we mix those? So we found that if he would mix purple flowers with the white flowers, the first generation crossed after them, what we call a hybrid. first hybrid generation, the F1 generation, he would get all purple flowers. Okay. But then if he took that F1 generation and mixed those, so he took some pollen from an F1 generation plant and mixed it with the female part, the stamen, I'm sorry, not the pistil on the female part of plant, fertilized those, used F1 plants to fertilize other F1 plants. Then he got a mix. About 3/4 of plants would be purple. About 1/4 would be white. So for every 3 purple plants, you would get 1 white plant. When you took the F1s, mixed them with the F1s, you get the F2 generation, the second hybrid generation. You get about 75% purple, 25% white. Right? So just labeling this here, yeah, he prevented the, again, remember these things want to self-fertilize. So he would go through and remove the male parts before they would mature, and then he would take, so he could actually control which plants spread with which plants. He would breed white flowers, purple flowers. That gave him F1, all purple. And the second generation, mostly purple, but a mixture. Okay. So this is what we see happens. So the P generation, the genotypes here, we'll say capital V for violet and lowercase v for white. Okay? So capital V for violet, lowercase v for white. The genotypes in the parental drift generation. In the phenotypes, what we actually observe, the violet and the white. In the second generation, the F1, we observe they're all violet, they're all purple. What's actually happened is the violet plants will contribute a violet allele. The white plants will contribute a white allele. Because the violet is dominant over the white, that's what we see, is we see violet. In the F2 generation, this is what we understand today that occurred. Mendel was getting really close to trying to figure out why. What we understand today, what occurred is you had some plants, so in the F1 generation, all of the F1 generation could give either a violet or a white allele. So sometimes they would give, the F2 generation, some of them would both receive a violet from both parents. Some of them would receive a violet and a white. And some of them would receive 2 whites. So both this and this, both the homozygous dominant and the heterozygous dominant, both of those would display phenotype. What's actually observed and displayed would be violet. Yes?
You always use the same letter for both traits, just different case.
If at all possible. If it's a single, if there's only 2 options, yes, if at all possible, we use the same letter. And use the lowercase and the uppercase. Which one's dominant, recessive. Now, what we run into, and we're going to see in a second, we run into things where it's not just 2 alleles, and it's not just dominant recessive. There are other possibilities. And then it gets more complicated. But if we keep it simple, yes, same letter. Yeah. And Mendel chose 7 traits that were all simple. They're all this or that, and one's dominant. So we're going to use that in all. Again, the phenotypes we observe violet and white. Both these, just fewer of the whites. And again, what we understand today, this just tells us that the violet is the dominant of the 2 traits. So if both are present, if you've got a violet and a white allele present, the violet will display over the white. All right, so how do we keep track of that? How do we figure this out? In section 1 of your text talks about probability, and that's perfectly good and just fine. I personally think we're probably just as good to just talk about the basic Punnett Square, right? So just grabbed from quotations there, and I've cited that as directly from Wikipedia. I just grabbed their description of what is a Punnett Square because that's better than I'm going to write it out. So let's just use theirs. They're telling us, Wikipedia is suggesting that a Punnett Square devised by British, actually that might be, I think that's from your text. That's directly from your text. I apologize. The picture's from Wikipedia. So your text says, Punnett Square devised by British geneticist Reginald Punnett can be drawn that applies the rules of probability to predict possible outcomes of a genetic cross or mating and their expected frequencies. Punnett squares are pretty easy to draw. We do need to understand the basic rules. So let's draw a couple Punnett squares for what Mendel was observing. Okay. The way you draw a Punnett square, you draw, if it's just a simple one trait Punnett square, I'm going to leave room above and to the side here to be able to add information. But you just draw a simple 2 by 2 square. And you put on, across the top here, you put the alleles for one parent. So in the F1 generation, again, we're crossing violet plants that have only been bred with other violet plants, so they're always just going to have violet alleles. And on the other side, so across the top, you put the alleles of one parent. On the side, you put the alleles of the other parents, in this case, lowercase v's, because those are whites. And you just go through here. So right here in this box, I'm going to take the B, the capital B from the top there, from this parent, and those together. So the genotypes, that tells us the genotypes for this particular crossing of the F1 generation, and they all end up, because in this box here, I'm going to take this capital B here, and this lowercase b. The same thing, I'll come down for the other possible crossing, I'm going to take this b, put it down here, this lowercase b here, and this capital B comes down, and this lowercase b comes across. Kind of like a multiplication table, if you remember those from elementary school or whenever you saw multiplication tables. Same basic idea, just a little simpler, except for using letters. So again, this is for the F1 generation, the one where we saw all of them were purple, because that was the only possible combination. Is B's from one, capital B's from one, the violet parent, lowercase b's from the other parents. The capital B's will always, because they're dominant, capital B is the one that will this way. Now the interesting one, this one's not too interesting. Here's the interesting one. The F2 generation, we're going to take all of these, so all of these F1s have got one purple allele and one white allele. The F2 generation, so well, how did that mix up? So we put the alleles of one parent across the top. And again, every F1 generation has got a capital B and a lowercase b. So this parent's going to have a capital B and a lowercase b. This parent on this side is going to have a capital B and a lowercase b. And again, what this does is it allows us to visually see what the rules of probability are telling us. Again, end of section 1 talks about how the rules of probability apply here. I just find this is an easier way to try to digest that, in my opinion. So about 25% of the offspring are going to have a capital V from one parent and a capital V from the other parent. Another 25% are going to have capital V here and a lowercase v. Another 25% are going to have a capital V from this parent and a lowercase v from this parent. And the last 25% are going to get a lowercase v from here and a lowercase v from here. So about 25%, 1/4 here, about 25% are going to have capital V's from each parent, homozygous dominant. 25% here and 25%, a total of 50%, are going to have one capital V, one dominant allele, and one lowercase v. So a total of 75% are going to display the dominant trait. 25% here, 25% here, 25% here, about 75, 3 for every one that's going to display the two lowercase v's. It's going to be homozygous recessive. It's going to be the white plant. So Mendel found about 705 violet flowers to about 224 plants with white flowers. And then your text, I just pulled this data from your text. As best I can tell, this is Mendel's original data that he recorded over about a 10-year period. In his F2 generation, you can see, right, he's saying for just what we just saw there, Mendel was able to measure against 705 violet flowers for 224 white flowers in the F2 generation, just a little over 3 to 1. A little more than 75% to 25% violet to white, which is what we would expect based on this Punnett Square. And we look at these other 6 traits that he looked at. Okay, we talked about the seed color, yellow versus green earlier, right? And again, if the parent generation are yellow seeds and green seeds, okay, and he mixed those because the yellow is the dominant. In the first hybrid generation, they're all carrying one yellow and one green allele. In the next generation, when we take these that have got one yellow and one green, then we'll see this breakdown of about a 3 to 1 mix between the yellow to green, about 3 times as many yellow as there are greens. And we see the same pattern fall, just about 3 to 1 for all 7 of these traits. Consistent, consistent, consistent. That tells us we've got something. This is important. Consistent result, repeatable over different scenarios. And again, so he does this in the mid to late 1800s. I think he's conducting these experiments, if memory serves, in the 1850s and the 1860s. And it's about the early 1900s before it actually gets uncovered and supported, because we got to defeat this prevailing scientific theory of blended inheritance. Okay. Now, Mendel only studied traits where there were only 2 alleles. One of those 2 alleles is dominant. And that occurs in a number of different traits across a number of different organisms, right? That is not an uncommon scenario. There are a lot of things we can point to. Nothing just off immediately top of my head, at least not humans. I want to talk about cattle, and because I grew up raising cattle, I know a few things there. But for example, I pulled this from a colleague of mine. 2 examples we can see is what we call codominance. That's where you've got 2 different alleles, but they're both completely dominant. Right? And I'm not an expert in chickens. Let's say if we have in chickens where you've got a black trait, a black allele is codominant with a white allele, that means if I've got one of each, right? If I'm homozygous white, the chicken will be white. If I'm homozygous black, the chicken will be black. If I've got one allele of each, I'm going to have some variation in that chicken. Some white and some black feathers. Refer to that as speckled. Sometimes you get what we call incomplete dominance. And that's where in the heterozygote, the one that carries one of each allele, you still can tell that it's red. You can still, in this case, you got red, white, and pink. In this case, you can still see that there's a red allele there. There's a red allele there. It's just because there's only one red allele, it's not getting expressed quite as vibrantly. I do seem to remember that does occur in a fair number of plants. The flowers that Mendel was observing in his peas did not exhibit this incomplete dominance. They were either purple or they were white in general. That made his findings a lot more useful. But there are a number of genes we can encounter where we have different types of inheritances. It doesn't just have to be dominant and recessive. We see this in blood types in humans. I have mentioned before, to the best of my knowledge, I'm type O. My dad told me he's type O. My mom's type A. Right? So the way that works, you have 3 alleles. Now, positive and negative, that's another thing. Okay, that's another gene on another chromosome. But we're just talking about the letter, the A and the O and the B. That is a single gene where there are 3 different alleles. You have the A allele, you have the B allele, both of which are dominant to the O blood type. But A and B have the same level of dominance. So you do have 3 blood types. You have you have 4 blood types. You have A, B, AB, and O. Those are the phenotypes. The genotypes can be, if you're A, that means you either have 2 A's or you have 1 A and an O. Your blood type A. So your phenotype is A, your genotype could be A, homozygous A, or heterozygous A. If you're B, if you type B, you either have 2 B's or 1 B and an O. If you type AB, that means you got an A from one parent, a B from the other one, and they're codominant. Type O, that's only going to occur if you got a type O allele from each parent, because it's the recessive. So it's only going to display if it's homozygous. That's how all recessive traits are going to be. They're only going to display if they're homozygous. Questions here? And what that means is it just means that your red blood cells have got a particular antigen on their surface. Right? The O blood type has no antigens. And that tells us whether which blood types can donate to which blood types. Depends on whether the body will reject or utilize the antigens or not. Okay. All right. Questions, comments, complaints for me? Let's see. I don't think that's the end. I think I've got a few more things. Entire chromosomes determine the sex within, at least for sure in humans. Now, some species it's a little more complicated than this, but in humans it's pretty straightforward. One of your pairs of chromosomes, humans have got 23 pairs of chromosomes. One of those pairs of chromosomes is what we call the XY chromosomes. So males will have, females, we'll start with those. Females will have 2 full-sized X chromosomes. In human species and many other species, females will have 2 full-size X chromosomes. It's one of their 23 pairs is the X chromosome, a pair of X chromosomes. In males, all the cells in a male body will have 1 full-size X chromosome, and then we'll have 1 kind of half-runted Y chromosome. It's not a full X chromosome. Part of it is homologous to the X chromosome, so they will pair up. The X and the Y will pair up during meiosis 1. In some species, there is actually some crossover during meiosis 1 between the X and the Y, not a lot, because there's just not a lot of genetic information on the Y chromosome, okay? Because it's small, it's not full size. But what that means then, and you can see the Punnett Square here, simple Punnett Square. The female gametes, because the chromosomes divide up, female gametes will each, because the female only has X's to give, each female gamete, each female egg cell will have an X chromosome. Because the sperm cells, the male only has one X and one Y to donate, so half of the sperm are going to get an X and half the sperm are going to get a Y. And the presence of a Y chromosome will determine if the offspring is male or not. It's a genetic disease where some males will actually receive 2X chromosomes by just through genetic checkpoint during meiosis metaphase 1, where one gamete can actually end up with 2X chromosomes in it. So there are human males on occasion that will have 2X chromosomes in a single Y, and that is they tend to have a number of troubles. They're typically infertile, but you're welcome to look up that information. Real quickly here, there was a question last time about twins. The question about twins. We talked about, yeah, you had a question about twins. I used to do a little research. Identical twins, not genetic, just pure circumstance, spontaneous. Fraternal twins, where you have two different egg cells get fertilized by two different sperm cells. And those are no different than any other pairs of siblings. They're just born at the same time. That is genetic. We call it hyperovulation. So that is actually that there is a gene that will code 2 eggs. Yes, female will ovulate 2 eggs at once, and that is actually genetic, and that's true. And I mentioned in sheep. That's true in sheep. It's true in humans. Most mammalian species, hyperovulation is a genetic trait that can get passed on. I believe it is dominant in most species. I know in sheep it's dominant. I was able to find that information.
Thank you.
You're very welcome. No problem. Other questions? Awesome. Let's call that a day. We'll go to the lab. I'm going to let this video save, and then I'll upload it to YouTube.